Welcome back to the deep dive. Today, we are undertaking a structural deep dive into, well, one of the most mechanically and chemically complex organs in the human body, the ear.
It really is. I think we often just think of it in terms of sound, but it's this amazing, highly specialized, three-chambered marvel.
And it's doing two huge jobs at once, right? It's managing both audition, which is hearing, and equilibrium, which is our sense of balance.
Exactly. And they're fundamentally integrated. So when you look at the histology, the actual microscopic blueprint of the ear, you realize just how dense and high performance this system is.
So our goal today is to kind of trace that path. We want to follow sound waves and, I guess, gravitational signals from the moment they enter the head.
All the way through. Through the different anatomical chambers,
Yeah.
right to the specialized mechanoreceptors that convert all that mechanical energy into electrical impulses.
We're going to unpack this step by step so you can walk away with a really crystal clear understanding of what makes this tiny organ tick.
Right. And to structure that, it helps to visualize the ear as three distinct interconnected divisions.
Okay, what on?
You've got the external ear and the middle ear. You can think of those as really purely mechanical systems.
So they're the scaffolding in the engine room.
Oh.
They collect, funnel, and dramatically amplify all the acoustic energy.
Precisely. And then you have the internal ear.
And that's the sensor array.
That's the sensor array. It houses all the true sensory receptors, taking those amplified mechanical signals and converting them into electrical signals the brain can actually understand. It's the sophisticated core for both sound and movement.
Before we jump into those structures, I think it's worth just marveling at its origin story. The ear has a surprisingly diverse embryological background, which kind of hints at why its final structure is so complex.
It's a fantastic origin story, and the most critical part, the internal sensory part, actually develops first.
Really?
Yeah, early in gestation, end of the third week, you get this thickening of the surface ectoderm near the developing brainstem. It's called the otic placode.
And this placode,
Yeah.
it doesn't just sit there.
No, it quickly folds inward, it invaginates, and by the fourth week, it literally pinches itself off from the surface to form the otic vesicle, or the otocyst.
So the essential sensory structure, the part that's going to eventually detect sound and gravity, is literally a piece of surface skin that just decides to dive inward and burrow into the developing head.
That's the foundation of the membranous labyrinth. That otocyst sinks into the surrounding mesenchyme, and it becomes the primordium, the blueprint for all the specialized epithelia lining the entire system.
And then the rest of the ear is sort of built around this central sensor.
Exactly. The surrounding structures, the collection and conduction pieces, they come from the pharyngeal arches, those structures that help define the head and neck.
Okay, so where does the middle ear come from?
The middle ear cavity and its lining. That comes from the endoderm of the first pharyngeal pouch. It forms something called the tubotympanic recess.
And that recess becomes what?
It ultimately provides the epithelial lining for the auditory tube, the eustachian tube, and the whole tympanic cavity.
And then on the outside, the corresponding groove from that arch, the first pharyngeal groove, grows inward.
Right, and that's ectoderm. And it forms the external acoustic meatus, which is the tube leading to the eardrum.
And you can't forget the little bones, the ossicles.
The transmission chain, those develop from the mesenchyme of the pharyngeal arches. So you have the malleus and indices from the first arch and the stapes from the second arch. It's a whole assembly project.
Surface tissue, internal pouches, cartilage, scaffolding,
Yeah,
all coming together inside the temporal bone.
exactly.
That structural assembly really sets the stage.
Right.
So let's start with the outside world, the external ear. We'll begin with the part we see, the auricle, or pinna.
The auricle is, the appendage projecting from the side of the head, and its really complex, characteristic shape is supported entirely by a structure of elastic cartilage.
And histologically, it's pretty simple. It's just thin skin covering that cartilage, and it has all the usual things, hair follicles, sweat glands, sebaceous glands.
Right. And while it's kind of vestigial in humans, compared to animals that can swivel their ears, it still plays that really foundational role in collecting and directionally focusing sound into the ear canal.
That canal being the external acoustic meatus.
Yep. It's about 25 millimeters long, so just under an inch, and it's purely an air-filled gateway.
So its main job is just to conduct sound to the eardrum. Simple as that.
That is its primary job, but it's not just a passive tube. It's actually a bit more clever than that.
Oh.
Because of its length and shape, the meatus acts as a resonator. This is a subtle but really important amplification step.
So it boosts the sound.
It enhances the sound pressure, reaching the tympanic membrane by about a factor of 2. And it does this specifically for frequencies in that crucial 2,000 to 5,000 hertz range.
which is right where human speech tends to fall.
Exactly. It's a built-in frequency booster, so it starts the amplification process before the sound even hits the first membrane.
That's fascinating. And the structure of the canal itself, it changes along the way, doesn't it?
It does. It has a mixed structure. The lateral one-third, the part closer to the outside, is supported by cartilage. But the medial two-thirds are actually carved out within the temporal bone itself,
And it's all lined with skin.
all lined by skin that's continuous with the auricle.
And it's in that skin that we find this unique specialization that, well, often leads to clinical trouble. The production of cerumen or earwax.
This is a great example of histological specialization meeting a unique environmental need. The skin in that lateral cartilaginous part of the meatus has hair follicles and sebaceous glands, but it also has these highly specialized ceruminous glands.
What are those like?
They're coiled tubular glands, and they look a lot like the apocrine sweat glands you'd find in, say, your armpit.
And the cerumen is a mix of all these secretions plus shedding skin cells. But why? Why does the ear need this specialized cleaning system?
Because the external acoustic meatus is structurally unique. It's the only blind pouch of skin in the body.
A blind pouch.
Yeah. Skin normally sheds cells outward, right, into the environment. But here, those cells have nowhere to go but inward, and they could potentially pile up against the eardrum.
So the cerumen production is the body's self-cleaning defensive mechanism. It works like a tiny internal conveyor belt.
Precisely. The cerumen traps those desquamated cells, it lubricates the skin, it impedes foreign particles like dust or even insects, and it provides some really essential antimicrobial protection.
And when that system fails,
You get impacted cerumen. And if it gets bad enough to plug the meatus, you immediately get a specific type of hearing loss, a conductive hearing loss, because you're physically blocking the sound waves from ever reaching the eardrum.
okay, so that moves us logically to the target of that sound wave, the middle ear. This chamber is often called the mechanical transformer.
It is, because its whole job is to bridge the air-filled external ear with the fluid-filled internal ear.
And that transition is absolutely critical.
It's everything. Because fluids and air have such different densities, if sound waves in the air just hit the fluid directly, 99.9% of the energy would just reflect back. You wouldn't hear anything.
So the middle ear, this air-filled tympanic cavity in the temporal bone, has all the equipment to overcome that mismatch.
Right. It has the ossicles, the regulatory tube, the muscles. Everything is designed to solve that density problem.
Let's just quickly set the scene. What are the boundaries of this little cavity?
Okay, so visualize it. Anteriorly, a thin layer of bone separates it from the internal carotid artery. Posteriorly, it connects to the mastoid air cells. The roof separates it from the brain's middle cranial fossa, and the floor separates it from the internal jugular vein.
So it's a very tight, very vital little space.
Very.
And the two most important boundaries for actually transmitting the sound.
That would be laterally the tympanic membrane, the eardrum, the point of entry, and medially the bony wall of the internal ear, which has the two critical openings, the oval window and the round window.
Let's focus on the eardrum for a second. The tympanic membrane. It sounds so delicate.
It is, but it's also structurally robust.
Yeah.
It's only about 0.1 millimeters thick with a surface area of about 65 square millimeters. And it's shaped like a little irregular flat cone.
With the apex, the umbo, attached to the malleus bone.
Exactly. And that concavity and its slight tilt are what give it the characteristic look during an exam.
Right, with an otoscope. The doctor sees that semi-transparent, light gray membrane.
And the light from the otoscope reflects off that conical surface, creating a visual landmark called the triangular cone of light, or the light reflex.
And if that reflex is distorted, that's usually a bad sign.
It often is. It can be a sign of pressure or fluid buildup behind the membrane.
Now, histologically, it's a three-layer sandwich structure. Let's build it from the outside in.
Okay, so layer 1, the outer layer, is skin. Stratified squamous epithelium continuing from the ear canal. Layer 3, the inner layer, is the mucous membrane facing the middle ear. A simple, low cuboidal epithelium.
And the really crucial middle core that gives it its tension.
That's the connective tissue core. It's dense, irregular connective tissue, and the fibers are arranged very strategically. You have an outer layer of collagen fibers running radially, like spokes on a wheel.
And an inner layer running circumferentially, like a tight belt.
Precisely. That architecture is what gives the main part of the membrane, the pars tensa, its essential tautness.
Okay, so you have the pars tensa, the tense part, which is the large lower area.
Yeah.
And then there's the other bit.
The small upper region is called the pars flaccida, the loose part. It's superior to the malleus, and it lacks that prominent fibrous core, so it's much more flexible.
And much more vulnerable to things like retraction from pressure changes.
Exactly. Now, clinically, any perforation or rupture can cause immediate noticeable symptoms.
I'd imagine.
Absolutely. You get an open connection between the outside world and the middle ear. That leads to pain or otalgia, drainage, otorrhea, ringing, tinnitus, and sometimes even vertigo. It almost always results in some degree of conductive hearing loss.
Okay, so past the membrane, we get to the central players of sound transmission, the three auditory ossicles.
The malleus, incus, and stapes, they're suspended across that air-filled cavity, connected by tiny movable synovial joints.
And they don't just bridge the gap. They act as a carefully calibrated lever system.
Yes. Designed to maximize the force that gets transmitted to that tiny oval window.
So let's walk through the chain.
The malleus, the hammer, is attached to the eardrum. It articulates with the incus, the anvil, which is the largest of the three, and the incus links to the final piston in the system.
The stapes or stirp.
Right. And its footplate is minuscule, only about 3 square millimeters, and it fits perfectly into the oval window, ready to generate those fluid waves in the cochlea.
This mechanical chain is so delicate that when it goes wrong, the functional impact is huge. A major example is otosclerosis.
Otosclerosis is a very common cause of acquired hearing loss. It affects up to about 1% of the population, clinically.
And what's actually happening at the tissue level.
You get this abnormal, localized overgrowth and remodeling of bone, specifically right around the oval window.
How does that happen?
Well, you see a localized area where osteoclasts are aggressively removing mature bone, and then it's replaced by thicker, structurally immature, what we call woven bone.
And this abnormal bone grows around the stapes.
It essentially encases the tiny joint that holds the stapes in the oval window.
So if the stapes needs to vibrate like a piston and it gets encased in abnormal bone,
It becomes fixed, frozen,
Ah.
and that fixation just stops sound energy from being transmitted into the fluid of the inner ear. The result is a profound conductive hearing loss.
but it's often treatable.
Highly treatable. Often. The modern treatment is often a stapedectomy where the fixed stapes is removed and replaced with a tiny prosthesis. It can be incredibly effective.
That example really perfectly illustrates the difference between conductive hearing loss and the other major type. Can we just quickly delineate those two categories?
Sure, so hearing disorders fall into 2 major bins. Conductive hearing loss is mechanical. Sound waves are physically blocked or dampened before they reach the sensory receptors.
So that would be impacted cerumen, fluid from otitis media.
Exactly. Or ossicle diseases like otosclerosis. This type is generally treatable if you can address the physical blockage.
And the other, often more permanent type.
That's sensorineural hearing loss, which accounts for something like 90% of all hearing impairment. This involves injury to the sensory apparatus itself,
So damage to the hair cells, the cochlear nerve.
or the CNS pathways, or even the auditory cortex. The causes are things like meningitis, acoustic trauma from noise exposure, certain ototoxic drugs, or physical trauma.
And we have to mention the type associated with aging.
Presbycusis. This is a sensorineural loss from aging that characteristically starts in the basal turn of the cochlea, which means the first frequencies to go are the high frequencies.
For profound sensorineural loss, the textbook mentions cochlear implants.
A cochlear implant is an electronic marvel. It bypasses the damaged hair cells completely. It has a microphone and processor outside, and an internal receiver connected to a multi-electrode array,
And that array is surgically inserted right into the cochlea.
right along the wall of the cochlear duct, yeah. And it stimulates the cochlear nerve endings directly, essentially taking over the job of the destroyed hair cells.
Amazing. Okay, let's go back to the middle ear's structures for a moment.
Yeah.
How does the system regulate itself?
It uses 2 tiny skeletal muscles to provide protection through what's called the acoustic reflex or attenuation reflex.
Okay, what are they?
You have the tensor tympani, which attaches to the malleus and increases tension on the eardrum. And then the stapedius muscle, famously the smallest skeletal muscle in the body, which attaches to the stape and dampens its movement.
And their job is to basically clamp down when things get too loud.
Exactly. It's an involuntary contraction that stiffens the whole ossicle chain to reduce vibration. It protects the delicate inner ear from sustained loud noises.
But you have to remember, the reflex is slow.
It is. It takes milliseconds to engage, so it offers almost no protection against a sudden impulse noise like a gunshot.
Right. Now, what about air pressure? That's the auditory tube or eustachian tube.
This is a narrow channel about 3.5 centimeters long, connecting the middle ear to the nasopharynx. It's lined with respiratory epithelium.
And its job is twofold, right? Equalize pressure And drain secretions.
Precisely. Pressure equalization is why your ears pop on an airplane. The tube is normally closed, but it opens when you yawn, swallow, or chew.
And that venting is essential for the eardrum to vibrate properly.
It is, and this structure is the key to understanding why ear infections, otitis media, are so common in little kids.
Because their anatomy is different.
It is. In children, the auditory tube is shorter, narrower, and much more horizontal than in adults. So it's a much easier path for infections from the pharynx to travel up into the middle ear.
And there's also the tubal tonsil nearby.
A small mass of lymphatic tissue, yeah. Also, one last detail here. The mastoid air cells.
The air spaces in the bone behind the ear.
Right. They extend from the middle ear cavity, and because they're lined by a continuous epithelium, an infection in the middle ear can easily spread into them, causing mastoiditis.
Okay, let's wrap up the middle ear by revisiting its mechanics. The total acoustic energy is amplified roughly 60 times. How does that happen?
It's achieved by three factors working together, and the whole point is to overcome that massive impedance mismatch between air and fluid.
And the biggest factor is just the difference in surface area.
That's the main booster, absolutely. You're taking the vibrational force collected over the large area of the eardrum, about 65 square millimeters, and concentrating it all onto the tiny area of the stapes footplate, just 3 square millimeters.
So that concentration of force results in a pressure increase of about 22 times.
Exactly. Then you have the mechanical advantage from the ossicle chain itself. It's a classic lever system.
Because the malleus is slightly longer than the incus.
Right. That arrangement multiplies the force by about another 1.3 times.
Okay, so 22 times from the area times 1.3 from the lever, and then the contribution from the external ear.
The resonance we talked about in the external acoustic meatus, that contributes an amplification factor of about 2 times. So you calculate 2 times 22 times 1.3, and you get
Nearly 60 times. The necessary boost to launch a hydraulic wave into the cochlear fluid.
that's it.
And now we make that transition into the internal ear, the true sensory apparatus.
Right. And this is structurally defined by 2 systems nestled within each other, the bony labyrinth and the membranous labyrinth.
So the bony labyrinth is the series of cavities carved into the temporal bone. And inside that is the membranous labyrinth.
A continuous system of sacs and tubules made of soft tissue and epithelium, which contains all the sensory cells.
And these labyrinths are defined by 3 distinct and chemically unique fluid spaces. This is critical.
The chemistry is the energy source. First, you have the endolymphatic space. This is the fluid inside the membranous labyrinth.
And it has a composition like intracellular fluid.
Exactly. Very high potassium, or K plus A, and very low sodium, Na plus A.
And where does this specialized fluid come from?
It's produced by the highly specialized epithelium of the stria vascularis in the cochlear duct. You can think of the stria as the ear's tiny essential potassium battery, and it drains via the endolymphatic duct.
Okay. Then you have the fluid that surrounds the membranous labyrinth.
That's the perilymphatic space, filled with perilymph. And perilymph is chemically the opposite. Low K plus E, high Na plus E. It's a lot like extracellular fluid or cerebrospinal fluid.
And where does that come from?
It's an ultrafiltrate from the microvasculature lining the bone, and it drains via the cochlear aqueduct into the CSF.
So that dual fluid system, high potassium inside, high sodium outside, is the engine that powers the hair cells. And there's one last localized fluid space.
That's the chordolymphatic space. It's the intercellular space within the tunnels of the organ of Corti, and it contains chordolymph, which is basically the same as perilymph.
So maintaining that huge electrical gradient created by the high potassium endolymph is everything.
It is the fundamental prerequisite for hearing and balance transduction.
Okay, let's define the physical structures of the bony labyrinth, starting with the central chamber.
The central oval chamber is the vestibule. It's where the stapes inserts at the oval window, and it contains the recesses that house the utricle and saccule.
And extending from that are the structures for rotational movement.
The 3 semicircular canals, anterior, posterior, and lateral, they're positioned at right angles to each other, which is structurally brilliant.
Because that allows them to sense movement in all 3 dimensions.
Exactly. Sagittal, frontal, and horizontal planes. They each expand into an ampulla where they meet the vestibule.
And finally, the auditory structure, the snail shell.
That's the cochlea. It's a cone-shaped helix that makes about 2.75 turns around a central core of spongy bone called the modiolus.
And the modiolus is essential because it houses the nerve ganglia for hearing, the spiral ganglion.
Right. And suspended within all of these bony structures is the membranous labyrinth.
Which consists of two continuous divisions.
You have the vestibular labyrinth, which includes the 3 semicircular ducts inside the bony canals, all continuous with the utricle, and then you have the saccule.
And an auditory component.
The cochlear labyrinth, which contains the cochlear duct, or scala media. And that's continuous with the saccule. These membranous structures house the 6 specialized sensory regions.
These 6 regions are where the magic happens. Let's list them again.
You have 3 cristae ampullares, 1 in each semicircular duct, which sense angular acceleration or head turning.
Okay.
You have 2 maculae, 1 in the utricle and 1 in the saccule, which sense static head position and linear acceleration.
And number 6.
The spiral organ of Corti in the cochlear duct, which is the dedicated sound receptor.
The mechanism that unites all 6 of these areas is the function of the hair cell, the true epithelial mechanoreceptor.
The hair cell is a spectacular example of mechanoelectrical transduction. Its top surface has this rigid bundle of projections, the stereocilia, arranged in rows of increasing height.
And the physical movement of these bundles is what determines the neural signal. And they have a specific directionality or polarity.
Yes. In the vestibular hair cells, there's a single true cilium called the canacilium, positioned just behind the tallest row of stereocilia, and the orientation relative to this canacilium determines everything.
So let's get down to the nanoscale level of how this works.
Yeah.
It's all about physical tension.
It is. If the bundle moves toward the canacilium, it generates tension on these tiny protein strands called tip links.
And that tension physically yanks open the mechanoelectrical transducer MET ion channels near the tip of the stereocilium.
Exactly. And since the bundle is bathed in that high potassium endolymph, potassium rushes in, causing rapid depolarization. That triggers calcium channels at the base and then neurotransmitter release.
And if the bundle bends the other way?
Movement away from the canacilium releases the tension, the MET channel snaps shut, and you get hyperpolarization, which inhibits the signal. It's an immediate on-off switch dictated entirely by the direction of mechanical force.
The structure of those stereocilia is key. They have to be stiff.
They're built like micro-rigid rods. Their core is tightly packed actin filaments, cross-linked by proteins like fibrin, and, importantly, a spin.
And we know a spin is essential because mutations in the gene for it cause hearing loss and vestibular dysfunction.
Right. In mice with this mutation, their stereocilia are too floppy to work properly, and they exhibit spinning behaviors.
What about the actual channel itself?
The MET channel complex contains transmembrane channel-like TMC proteins, specifically TMC1 and TMC2. And in fact, mutations in TMC1 are a known major cause of deafness in humans.
And the tip link, the physical wire that opens the gate, is made of what?
It's a fibrillar cross-linked composed of specific proteins like cadherin 23, CDH23, and protocadherin 15, PCDH15. The physical force applied to these delicate structures is literally what allows us to hear.
Once that hair cell depolarizes, it uses a highly specialized connection to talk to the nerve fiber.
It uses ribbon synapses. This is a special type of synapse designed for rapid and sustained release of neurotransmitters, in this case, glutamate.
So it's like a high-speed conveyor belt for neurotransmitter release.
That's a great way to put it. When calcium floods in, the ribbon quickly delivers a huge pool of pre-primed vesicles to the membrane for fusion, ensuring the nerve can fire rapidly and continuously.
And there are 2 types of hair cells in the vestibular labyrinth.
Yes, type I cells are flask-shaped and are mostly enveloped by a large chalice-like nerve ending. Type 2 cells are more cylindrical and have more traditional bouton nerve endings at their base.
Okay, let's move to the balance sensors, starting with the cristae ampullares, which sense angular acceleration, head turning.
These are thickened epithelial ridges in the ampulla of each semicircular duct.
Yeah.
They house type I and II hair cells, and all their stereocilia are embedded in a large gelatinous cap called the cupula.
So when I turn my head, how does that become an electrical signal?
When you rotate, the bony labyrinth moves with your head. But the endolymph fluid inside the duct lags behind because of inertia. That relative motion, the endolymph dragging against the cupula, sways the cupula, and that deflects the embedded stereocilia, generating nerve impulses that correspond to your rotational speed and direction.
And this contrasts with the other pair of vestibular sensors, the maculae of the saccule utricle. They sense gravity and linear acceleration.
Right. The maculae are sensory thickenings positioned at right angles to each other. When you're standing, the utricle macula is mostly horizontal, and the saccule macula is mostly vertical.
And what makes these maculae heavy enough to respond to gravity?
They're covered by the otolithic membrane. And unlike the cupula, this gelatinous layer is weighted down by tiny crystalline bodies called otoliths, or otoconia.
Which are made of calcium carbonate.
Calcium carbonate and protein, yeah. And they are significantly denser than the endolymph.
So when I tilt my head, the mass of those tiny crystals is what does the work.
Exactly. Gravity or linear movement displaces this heavy layer of otoliths, causing the otolithic membrane to drag across and deflect the embedded stereocilia. That's what signals movement or tilt.
We transition now to the cochlear system, the dedicated receptor for sound. It all starts with the spiral canal being divided into 3 parallel channels, or scalae.
The center endolymph-containing channel is the scala media, or the cochlear duct. Above it is the scala vestibule, which contains perilymph and starts at the oval window.
And below it is the scala tympani, also perilymph-filled, which ends at the round window.
And those 2 perilymph channels, SV and ST, only communicate at the very apex of the cochlea through a small opening called the helicotrema.
The scala media itself is a triangular space, and its 3 walls define its function. Let's start with the upper wall.
The upper wall, separating the scala media from the scala vestibule, is the delicate vestibular membrane, or Reisner membrane. It's just 2 cell layers thick.
The lateral wall is that potassium battery we mentioned earlier.
The stapes vestibularis. It's a unique, highly vascularized epithelial structure that actively transports potassium ions into the endolymph, generating the high endocochlear potential that's necessary for transduction.
And the lower wall, the floor upon which the whole sensory system sits.
That is the basilar membrane. It's relatively flaccid, but critically, its physical structure changes along its length. It's narrow and stiff at the base, and it gets progressively wider and more flaccid toward the apex.
And that physical gradient is the key to frequency coding.
It is. And resting on that basilar membrane is the extraordinarily complex epithelial structure known as the spiral organ of Corti.
This organ contains the receptor cells.
Yeah.
You have a single row of inner hair cells and 3 to 5 ranks of outer hair cells.
Right. And the support network is incredibly intricate. You have pillar cells that form the triangular inner spiral tunnel.
And then the pharyngeal cells.
Right. The inner ones cradle the inner hair cells. The outer ones, or diater cells, support only the base of the outer hair cells, but send these long, thin processes up to form the reticular lamina.
And the reticular lamina is a key structural barrier.
It is. It's formed by tight junctions that act as a protective separation between the potassium-rich endolymph above and the low-potassium chordolymphatic space below. It makes sure only the tips of the stereocilia are exposed to that high potential.
And finally, covering the whole organ is the dectorial membrane.
A thick gelatinous sheet that projects over the hair cells. And crucially, the stereocilia of the hair cells are either embedded in or contact this membrane.
Okay. Let's tie all these parts together into the final act of sound perception.
It starts with the stapes pushing on the perilymph of the scala vestibule. This initiates traveling waves. These waves transmit through the vestibular membrane and propagate down the system.
But the key to extracting information is how this wave affects the basilar membrane.
The sound wave sets up a traveling wave in the basilar membrane. This is the physical basis of frequency discrimination, or how we perceive pitch.
And that's because of that structural variation, stiff base, floppy apex.
Beautifully simple mechanical principle. A sound of a given frequency will cause maximal vibration only in the specific region of the basilar membrane that resonates best with it.
High-frequency sounds vibrate the base. Low-frequency sounds vibrate the apex.
Exactly. And loudness is just encoded by the degree of displacement.
So that vibration has to be converted into a neuronal signal. How?
This is where the hinging matters. The basilar membrane, holding the hair cells, and the tectorial membrane, holding the stereocilia, are hinged at different points. So when the basilar membrane vibrates vertically, this difference creates a lateral shearing effect.
And that shearing force is what physically bends the hair cell stereocilia.
That's it. That deflection activates the ME channels, generates action potentials, and sends them off via the cochlear nerve.
Okay, let's follow those signals now with the innervation via the vestibulocochlear nerve, cranial nerve 8th.
CN8 has 2 distinct divisions. The vestibular nerve is for equilibrium. Its cell bodies are in the vestibular ganglion of Scarpa.
And these neurons collect information from the maculae and cristae.
Right. Their axons enter the brainstem, terminate in the vestibular nuclei, and then send secondary fibers out to the cerebellum and the nuclei that control the eye muscles. That's essential for coordinating balance with vision.
Then you have the cochlear nerve, the auditory division.
Its cell bodies are in the spiral ganglion of Corti, housed within the modiolus. And a remarkable 90% of its dendritic processes synapse with that single row of inner hair cells.
So the inner hair cells are the primary auditory sensors.
They are the remaining 10% synapse on the outer hair cells. These axons then bundle together, form the cochlear nerve, and ascend to the auditory cortex.
We also have to acknowledge the fibers that run from the brain to the ear.
Those are the efferent fibers that all of a cochlear tract. They carry impulses from the brain to the organ of Corti. The theory is that these fibers modulate auditory input, maybe filtering or suppressing background noise, to enhance the clarity of a signal.
Now for the clinical consequence of vestibular system failure. Vertigo.
Vertigo is that disabling sensation of rotation or spinning when you're actually still. It signals vestibular dysfunction.
And the most common mechanical disorder is benign paroxysmal positional vertigo, BPPV.
This is a classic example of mechanical misalignment. The heavy otoconia, the little crystals, detach from the utricle macula and drift into one of the semicircular ducts.
So when a person moves their head, those tiny detached stones move inside the fluid channel where they shouldn't be.
Precisely. The movement of these free-floating otoconia causes inappropriate fluid movement, which deflects the cupula and tricks the brain into perceiving massive spinning.
And what about the chronic disease related to fluid pressure, Meniere disease?
Meniere's involves a triad of symptoms: dizziness, tinnitus, and low-frequency hearing loss. It's caused by a blockage that prevents the normal drainage of endolymph,
Which leads to increased pressure and distension of the membranous labyrinth.
a condition called endolymphatic hydrops. That increased pressure disrupts the delicate fluid chemistry and the mechanical motion of the hair cells.
To conclude our structural journey, we have to highlight the unusual and vulnerable blood supply to the internal ear.
Right. The external and middle ears are served by the robust external carotid system. But the delicate membranous labyrinth, the sensory core, is supplied by a single intracranial labyrinthine artery.
And what makes this supply so precarious?
It's a terminal artery. It has no anastomoses, no safety net of backup connections. If that artery becomes blocked, the internal ear is highly susceptible to immediate ischemia and functional loss.
What an elegant system. We've traced sound and movement through 3 stages. Capture by the external ear, massive mechanical amplification by the middle ear,
And finally, conversion into hydraulic waves in the internal ear fluids. And we've learned that conversion relies entirely on the differential stiffness of the basilar membrane for pitch.
and the shearing force that deflects the hair cell mechanoreceptors. That physical deflection opens the MET channels via the tip links, generating signals that travel along CN8.
Yes. So if you take away 3 essential points, remember the fluid chemistry, high K plus endolymph versus high NAN plus perilymph. Remember the mechanism of amplification, how the area difference is key.
And the distinction between hearing loss types. Conductive is mechanical blockage, while sensorineural is biological damage to the hair cells or nerve.
And if we leave you with one final provocative thought, just consider the sheer mechanical precision required for us to perceive our world. The reason you can hear a whisper or feel the slight pull of gravity in an elevator comes down to the bending of protein structures
The cadherin 23 and protocadherin 15 of the tip links.
that are only nanometers in size, physically pulling open an ion channel. The entire universe of audition and equilibrium is founded on that instantaneous nanoscale physical event.
Absolutely stunning. Thank you for joining us for this deep dive into the histology of audition and equilibrium.